All right, thanks everyone for coming back for this morning session. I'm Nick Veomett, VP of Corporate Development at Artiva Biotherapeutics. Let's see, green is next. We're a publicly traded company. We'll be making forward-looking statements. So Artiva Biotherapeutics is a leading NK cell therapeutics company based in San Diego, California. We're focused on developing an off-the-shelf allogeneic NK cell therapy that's given in combination with monoclonal antibodies in autoimmune disease. We have proof of concept data, which I'll go into shortly, in multiple oncology settings, which provides proof of mechanism of what we're pursuing in autoimmune disease, as well as shows the flexibility of the modality. As I said, we have ongoing trials in autoimmune disease, in refractory RA, lupus, and other B-cell-driven autoimmune indications. We're taking a manufacturing-first approach. The company was actually founded in 2019 based on technology coming out of a Korean company, GC Cell, and they spent over a decade optimizing a process for scaling up and cryopreserving NK cells in a highly scalable and uniform way. And the process is such, I'll go into a little detail, that we see COGS in the commercial setting being less than $1,000 per 1 billion cell vial, which is the starting dose, and what it's vialed at. So very reasonable cost of goods for a cell therapy in a way that this could be given very broadly and in outpatient community settings. We IPO'd last year, so well capitalized, publicly guiding to have cash into the second quarter 2027. And we'll be sharing initial safety and translational data in our autoimmune indications this year, and then activity in our lead indication in the first half of next year. So what is Artiva? This slide represents sort of three pillars of our strategy. The first is the NK cell itself. So the diagram on the left shows allo-NK, which is a non-genetically modified NK cell given in combination with monoclonal antibodies. So since it's not genetically modified, it doesn't have any CAR targeting. So we're basically utilizing the natural mechanism of NK cells, which is antibody-dependent cellular cytotoxicity, or ADCC. So the picture on the left, the little gray, is the antibody. That's what's actually binding to the cell target. And then the NK cell is using the CD16 Fc receptor to bind the Fc portion of the antibody and potentiate ADCC activity. As I said, we take a manufacturing-first approach. We select NK cells from cord blood. This provides two advantages. One, we can actually select cord blood units with specific genetic attributes. And since we're not engineering anything into the NK cell, basically the genetics that we start with from the donor get carried through into the drug product. And importantly, one of the ones that the markers we look for is a high-affinity CD16 variant, which is a natural variant in the population. So that gives all of our drug product a high-affinity CD16 receptor for this ADCC mechanism. And then, as I said, the process is highly scalable. So we're able to generate from one cord blood unit thousands of these cryopreserved NK cell vials, which then leads to really our strategy is to bring these to the outpatient setting and the outpatient community rheumatology setting in autoimmune disease. It's very simple to administer. It's a 5 to 10 minute IV infusion. They're shipped as they're cryopreserved, so they can be delivered directly to the site. Very simple, thaw and infuse. So as I said, we have proof of principle data in 2 oncology indications. And so I'll admit, I think NK cells have gotten a bit of a bad rap. There's been mixed, sometimes negative results, especially with CAR NKs in the oncology setting. You might be forgiven to ask, this mechanism sounds very simple. You're just giving an NK cell with an antibody that's already used in this setting. How is that going to work? But there's actually data on over 80 patients across non-Hodgkin lymphoma and Hodgkin lymphoma that have been treated with non-engineered, non-targeted NK cells in combination with antibodies. On the top are actually academic studies out of University of Minnesota. On the left is NHL. On the right is Hodgkin lymphoma. And then on the bottom are our allo-NK studies. So the left in non-Hodgkin lymphoma was NK cell in combination with rituximab. And you can see the very high response rates and quite good durability. I'll show a little benchmarking on the NHL on the next slide. And then similarly in Hodgkin lymphoma, which was a combination of NK cells with asymptomig, which is a CD30 targeted NK cell engager. Again, showing high response rates and durability of response. So this provides proof of principle that this mechanism is actually able to drive deep depletion of the target cells and durable responses. This is digging into what was basically the bottom left panel, which is our study in non-Hodgkin lymphoma. It was allo-NK in combination with rituximab. The top represents the responses in CAR-T naive patients. So this is to be able to benchmark against what others have shown in this space. But these were late-line patients, median of 3 prior lines of therapy, all but one with aggressive lymphoma types, and all of them had been treated before at least once, sometimes multiple times, with rituximab in combination with other chemotherapeutics. So we're coming in again with rituximab plus our NK cell and seeing these high level and durable responses. And so you can see that that actually compares well with autologous CAR T cells as well as other allogeneic T cell approaches. So why am I telling you about oncology? I said we're focused on autoimmune disease, but really what we're trying to do in autoimmune disease is very similar mechanism of action. In oncology, you're using rituximab, the antibody, to target B cells and try to get rid of the cancerous B cells, both in periphery and in lymphoid tissues. And the evidence of that in oncology is you get a complete response if you're getting rid of the cancer cells in the lymphoid tissue. In autoimmune disease, we're essentially trying to do the same thing, drive deep B cell depletion in periphery, but importantly in lymphoid tissues, and then cause this sort of reset of the immune system where you get more naive B cells coming back, and you're able to drive durable remission or reduction in the disease burden. So we see a real advantage of allo-NK in being able to go into true community rheumatology settings. So this represents sort of a matrix where we're thinking about activity and tolerability. So T-cell based modalities, autologous CAR-T, now we're seeing some T-cell engagers, have shown great activity both in oncology and with emerging data in autoimmune disease across multiple B-cell driven autoimmune diseases. They're able to induce deep B-cell depletion, durable responses in some patients where responses are maintained well after the course of therapy. But anything T-cell based has the liability of cytokine release syndrome and neurologic toxicity, ICANS. NK cells work differently. So NK cells utilize more porphyrin and granzymes and a different mix of cytokines to signal, and they don't cause escalating CRS. They also don't expand in vivo, so you don't get the expansion and the CRS that can come with it. So we see the potential for having a much better tolerability profile. In fact, in our NHL study, we saw less than 10% of patients reported any CRS. They were all low grade, but in going back and looking at those events, patient by patient, all of them are reclassified as infusion-related reactions. Essentially, they occurred quickly after infusion. We didn't see any elevation of cytokines, notably IL-6, which you see with CRS and T cells. And so we see our therapy being able to actually be sort of plug and play with how rheumatologists manage disease in the community setting. Whereas with a T-cell based approach, if there's a risk of CRS, even if it's manageable with hospitalization and tocilizumab, it's something that has to be dealt with, whereas ours can be completely just given outpatient, patient goes home, and there's not the risk of that event developing. So we are active in multiple clinical studies in autoimmune disease, 2 company-sponsored trials and 1 IIT. On the top left, we were the first allogeneic off-the-shelf cell therapy to go into autoimmune disease by expanding our reach into lupus nephritis first, and then expanded that trial into SLE more broadly. In that study, we're looking at combination with rituximab or obinutuzumab, which is another CD20-targeted monoclonal antibody. The second one on the bottom left is an investigator-initiated study looking at multiple autoimmune indications where rituximab is either approved or used routinely in clinical practice. This is at a single center in Florida that's really a true outpatient rheumatology practice. They have an infusion center on site, and they're running the entire trial in that setting. So a patient comes in for outpatient infusions, goes home, everything is managed at that site. So this is really proof of principle that our therapy can be given in that setting, and it's allowed us to look for those types of sites for our internal clinical studies, and we're seeing a lot of excitement among sort of investigators at those types of community sites to be bringing this type of therapy to their patients. And on the right is our phase 2A basket study that's open. We're recruiting in rheumatoid arthritis, Sjogren's, myositis, and systemic sclerosis. Looking at indications with large market potential, as you can see with, for example, RA, but also where there's still substantial unmet need. But we really think our advantage of being able to go into the community setting and be sort of plug and play with how rheumatologists are managing biologic therapies currently will be a huge advantage in these types of indications. And then I think our experience with the two trials on the left also allowed us to have a very sort of differentiated protocol. We're able to continuously enroll, so there's no stagger within dose cohorts. There's no required hospitalization at all, even on first infusion or for observation. And it's a basket study, so we have some flexibility into which indications we can expand for further investigation. So maybe to leave a minute for questions, just conclude with Artiva as a leader in NK cell therapy, targeting large markets in the autoimmune disease space, but where there is still significant unmet need and we can have an advantage in that outpatient setting. We have, as I said, clinical experience to date across oncology that shows both activity and tolerability, and a scalable manufacturing process with very favorable COGS that will allow this to be developed into something that truly could go broad. So we'll take any questions, and thank you for the time.